Hello everyone, my name is Hanzala and today I'll be presenting my project on microbiome diversity and composition analysis between healthy and Crohn's disease patients using R. So a little bit of background, the human gut microbiome is a massive community of microorganisms and they live inside our digestive system. So these organisms play a crucial role in digestion, immune functions, and overall health. And when these communities are disrupted or abnormal, it can cause diseases such as Crohn's diseases or other types of inflammatory bowel diseases. So in this project, I'm going to be using a data set from this study, and I'm going to be examining the differences in microbiome composition between healthy and Crohn's disease patients. And I chose this study because it's a treatment naive data set, meaning data was taken from patients before any form of medication was given. So there are no confounding variables. So the overall goal of this project is to compare the gut microbiome between healthy subjects and subjects with Crohn's disease. And we're going to be doing this by measuring the alpha diversity, which measures the richness and evenness of microbiome species within a single sample. Secondly, we're going to be measuring the beta diversity, which measures how different or similar microbiome communities are between samples. And lastly, we're going to be looking at the taxonomic composition, which will be looking at the abundance of bacterial families and comparing them between each group. So this is important because understanding the microbiome differences between healthy and diseased individuals can give us a better outlook and understanding on how these patterns result in these diseases. And the reason why we're going to be using a bioinformatics approach is because the data set is too large to be analyzed using traditional culturing methods. Bioinformatics allows us to process the data by normalization, calculation, and manipulation, and analyze the data by making graphs for visual representation of large-scale data. And this allows the study to be reproducible and statistically interpretable, and it's just more efficient than using old traditional culturing methods. So the data set I mentioned is going to be from the Jevers et al. Study. This data set includes metadata, which basically describes the data, and also includes the microbial abundance of different bacterial families. Like I mentioned, this is a treatment naive data set, which provides more accurate representation of the differences between the communities. A brief workflow is we're going to be downloading, importing the data set, and then we're going to be separating the metadata from the taxa abundance columns. We're going to be constructing a phyloseq object. We're going to be normalizing the data set, and then we're going to be calculating the diversity metrics for alpha diversity and beta diversity. And lastly, we're going to be generating a plot for each one of the diversities and the taxonomic composition. So we're going to be using these packages in RStudio. Phyloseq is widely used to organize and manage microbiome data. ggplot2 is really good for making plots for visualization. Vegan is used to calculate alpha and beta diversity metrics. dplyr is used to filter and manipulate the data. Reshape2 is used to reshape the data from a wide to long format. This is mainly used for a stacked bar plot. And then readxl is used to read in the data from an Excel file. So the approach we're going to be using for alpha diversity, we're going to be normalizing the data, then measuring the Shannon index, and then we're going to be making the box plot to show variation alpha diversity, meaning species richness and diversity and evenness between groups of each gut location. For beta diversity, we're going to be normalizing the data, and then we're going to be using the Bray-Curtis dissimilarity to find the differences between samples based on composition, and then we're going to be using a PCOA plot to calculate the distance between samples. Points that are closer together have similar microbiomes. Points that are further apart have different microbiomes. Lastly, we're going to be plotting the taxonomic composition using a stacked bar plot. We're going to be calculating the mean abundance of each bacterial family per diagnosis group to show the differences between the groups. So we found that CD patients, meaning Crohn's disease patients, have a higher Shannon diversity mean than healthy patients, which means that they have a greater microbiome diversity and richness. We found that both groups had overlapping points in the PCOA plot, and there were no distinct clustering, which means that the microbiome communities of both groups are pretty similar. And then lastly, these are the bacterial families that we found that were different between the groups and our taxonomic composition stacked bar plot. However, we did not perform any statistical significance analysis, so we cannot say that these are concrete conclusions. So in this tutorial, it was shown how to import microbiome data, how to organize microbiome data using Phyloseq, and how to normalize microbiome data. We also showed how to calculate Shannon index for alpha diversity, Bray-Curtis similarity for beta diversity, and the mean relative abundance for taxonomic composition. Lastly, it was shown how to plot box plots, PCOA plots, and stacked bar plots. What I've learned from doing this is understanding the importance of a treatment naive data set, organizing large data sets like microbiome data sets, and the purpose and the process of normalizing these microbiome data sets. Also, I learned how to calculate diversity metrics like Shannon index, Bray-Curtis similarity, and how to plot a PCOA plot. These are all the references and the citations, also a link to the published study and the link to the dataset. Thank you guys for viewing my presentation, and that is the end.